All right, hello everyone. Thank you for joining us today for the first webinar in our In Silico series, co-hosted by the PETA Science Consortium International and the Institute for In Vitro Sciences. I'm Kyle Martin, a toxicologist with the PETA Science Consortium International, and I'll be co-moderating today's webinar series along with Christy Sullivan, the Vice President of Education and Outreach at the Institute for In Vitro Sciences. Today, we're kicking off a webinar series designed to be accessible and informative, hopefully for attendees of all experience levels. And to facilitate this, we've not only invited great speakers for our 101 module, but also made resources available, including a glossary of terms that will be running and be updated for every webinar, and other relevant materials like a reading list and additional kind of cheat sheet, which can be found on the same PSI website where the registration link is housed. Our first three webinars are an In Silico Toxicology 101 series comprising 3 webinars, and future entries in this series will delve into more complex topics such as methods development, case studies, and efforts towards method qualification for regulatory purposes. Please note that the slides are already uploaded to the website, so you can pull them and make notes. And the recording from today's webinar will be posted online at the link shortly after the webinar. All future materials from this series will also be available at the link as the series continues. Amy, if you'd move to slide 2. Thank you. So on screen, you'll see a QR code. We have a number of opportunities for interaction today throughout our webinar, primarily in the form of polls. To participate, you can simply scan the QR code on screen with your phone, and the polls will activate once we get to that point in the presentation. You can also go directly to your browser at slido.com and enter In Silico 101 after the hash. We hope you'll join us in that, and I'm going to leave this on screen for just a moment to let everybody get a chance to scan in that would like to. All right, let's move to the next slide, please. Thank you. We also wanted to ask you to save the date for our next webinars, which will be the 17th and 18th of December next week at 10 a.m. Eastern. The next 2 webinars will focus on developing, applying, and validating in silico methods. This will include discussions of data requirements, as well as strengths and limitations of specific tools and their use cases. Attendees will need to register for those webinars separately on the website. Those links are already live, and you can register for those at any time. Let's move to the next slide, please. Today, we have 2 speakers presenting on computational tools for chemical hazard characterization. First, we're going to hear from Victoria Hall, a senior bioinformatician at Inotiv, and then from Brad Reisfeld, a senior principal predictive toxicologist at Inotiv. Their presentations are designed to flow together, so I'm going to introduce them both now. Victoria is a senior bioinformatician at Inotiv, specializing in computational toxicology and new approach methodologies. She develops workflows for physiology-based pharmacokinetic modeling, curates high-throughput screening data, and analyzes variability in vivo reference data sets. Victoria is passionate about public outreach and collaborates with federal agencies to build confidence in alternatives to animal methods and develop open access computational tools. And Dr. Brad Reisfeld is a senior principal predictive toxicologist at Inotiv, where he works on projects related to computational toxicology and NAMs. He's board certified in toxicology and is a fellow of the Academy of Toxicological Sciences. Prior to Inotiv, he was a professor of chemical and biological engineering and public health at Colorado State University. Dr. Reisfeld was a Fulbright scholar and a visiting professor in Thailand, and later spent a sabbatical year as a senior visiting scientist at IARC in France and at EFSA in Italy. He's published a number of papers on computational toxicology and co-edited a 2-volume book dedicated to the subject. Outside of work, he lives on a farm in Colorado and, in his own words, enjoys raising goats and other critters. All right. We'll be having a Q&A session at the end of our second presentation. So if we'll hold questions until then. The webinar is scheduled for roughly an hour with plenty of time, hopefully, at the end of the second presentation for questions. Everyone will be muted until then, and you can type your questions into the questions module within the Zoom toolbar at the bottom of your screen. Please do indicate if your question is directed to a specific speaker, and please do not add questions to the chat. It's difficult for us to follow them, and we'll probably ignore them. We have enabled closed captions. You can turn this feature on or off according to your preferences at the bottom of your screen. And without any further ado, I'm going to go ahead and hand this over to Victoria Hall.
All right, let me share screen here. All right, can you see my slides okay?
Yep, looks good.
I will take, okay. So thank you for the introduction, Kyle. So as he said, I'm Victoria Hall and I'm a senior bioinformatician at Inotiv. Thank you to the PETA Science Consortium and IIVS for the opportunity to speak to you all today about computational tools for chemical hazard characterization. And this is part of our webinar series on In Silico Toxicology 101. So for an outline today, this webinar will consist of two parts. So the first part I will be giving, where I'll be discussing learning objectives, the different types of NAMs, how NAMs fit into chemical hazard characterization, and a brief history of in silico NAMs. Then my fantastic colleague, Brad, a senior principal predictive toxicologist at Inotiv, will present the second part of the webinar, giving an overview of in silico NAMs, as well as the purpose, inputs, and outputs of various types of in silico NAMs. We will then have a summary of our presentation followed by a Q&A. All right, so let's get started with part one. So after attending this webinar, our goal is for participants to be able to describe some current uses of in silico NAMs and the state of their acceptance for regulatory applications. We want you to be able to explain how in silico methods can complement in vitro and in vivo data for chemical hazard characterization. You should be able to define important terms used to describe various aspects of in silico NAMs, as well as list the purpose, inputs, and outputs and basic flow information for common in silico NAMs. So as Kyle mentioned, we want this to be as interactive as possible. We want to make sure that we understand what you're learning, what you're interested in, and so we incorporated some Slido polls and quizzes throughout the presentation. And so for those of you who use Slido, you're probably familiar with this, but for those of you who are new to it, you can either scan the QR code using your phone or you can join at slido.com. And so we would like to know what word or phrase you associate with in silico approaches or NAMs in general. And so from here, we can kind of get a general feel of our background and then see as the webinar progresses how folks are learning and how perspectives might change. All right, I'm seeing a lot of Q stars. First methodologies, modeling. Fear, I like that one. Interesting. We'll give it another 30 seconds or so to make sure everybody can participate. Okay, so I appreciate all of these responses. It looks like we have a lot of very technical answers, which is encouraging because it means that people are at least familiar with some of the NAMs. But some of them are a little bit more complicated. Like I saw fear pop up. I know that they are very difficult and it is new for a lot of people and they can be really challenging. I see trust up here, which is a good one because it is hard to build trust in some of these models, especially when they do seem to be black boxes, right? No idea, very fair, very valid. So let's see as we move forward with this webinar series if we can't maybe get even more specific with some of these terms or have some more positive terminologies rather than some that are maybe a little bit, more fearful. Sorry about that. There we go. Okay, so now let's get into the actual webinar. So I do want to point out that for the entirety of our webinar series, you're going to see words that are in orange and bolded. And what this means is that they are going to be defined in our supplementary glossary. So if you don't want to click through slides or there's a term that you think is important and you don't have time to write it down, we are going to give a glossary. These words will be in it. So when we say NAMs, we are talking about new approach methodologies, which is a broad term that is often used synonymously or interchangeably with non-animal methods. Your opinion on that may differ, I understand, but people do use it that way. but it's a broad term referring to any technology used to assess chemical hazard without using intact animals. And these methods aim to replace, reduce, and refine animal testing, the 3 Rs, in areas like drug development, safety assessment, and toxicology. And so NAMs, by and large, can offer faster, more human relevant, and potentially more cost-effective results. And so there are 4 categories of NAMs I want to at least introduce you to in this webinar. And the first one is going to be ex vivo methods. And so ex vivo methods measure biological effects by using living tissues or organs. Examples of this might be tissue explants or perfused organ models. And these are going to be essential in capturing key events in toxicity pathways that can lead or link the interaction of a chemical with a biological target to a final adverse outcome in a way that is going to be a step down from those animal methods, but still going to be very biologically relevant. There are then in vitro methods, which can measure biological effects by using human or animal cells. And so examples of this are cell-based assays, high-throughput screening assays like ToxPass and Tox21, or organ-on-a-chip models, which are gaining steam throughout the field. And so similar to ex vivo models, these are very useful for capturing those key events in toxicity pathways. There are in-chemical methods, and in-chemical methods directly measure the chemical reactivity of a substance, with acellular assays and protein plasma membrane tests being examples of these two. And so these are good at capturing kind of that first step or that molecular initiating event in toxicity pathways. And last but not least, in the topic of most of our webinar today, as well as the rest of our series, is going to be in silico methods. And so in silico methods are computational methods that predict the intrinsic properties or fates of a chemical using existing data and algorithms. Examples of which include quantitative structure activity models, read-across methods, molecular modeling, machine learning, and artificial intelligence. And these are really, they act in complement to all the other NAMs that we discussed because they can help fill in data gaps, they can be predictive in ways that you might not necessarily get with other methods, or they can add human relevance to methods that might not have them. And so where do NAMs fit into this larger question of chemical categorizing and describing chemical hazard? And so if you're here, then you likely know that most toxicology studies are traditionally reliant on in vivo data, right? So in vivo data, while the most biologically relevant, are going to be comparatively low high throughput. They can be high cost. There are, of course, some ethical concerns around their use, and there is sparse human data. And so while no one is denying the usefulness of animal data, we'll talk about how it acts in complement with some of these NAMs in a second, there are some downsides to using them. And so recently, within the past few decades, there has been a paradigm shift towards integrated approaches to testing and assessment. And when we say integrated approaches, we are talking about using different combinations of NAMs along with in vivo data to answer toxicological questions. And by using NAMs, you can have higher throughput outcomes, you can have lower costs, NAMs can be broadly applied, and you can have very targeted biological assays. And this paradigm shift is really focused on those 3 Rs. And so for the role of NAMs, you can use them for screening and prioritization. So you can have the fast assessment of large numbers of chemicals to find ones that need further testing. You can fill data gaps, particularly by using in silico predictions, which reduces the need for new tests. There are mechanistic predictions, which can hypothesize and predict how a chemical might cause toxicity. This one is, of course, a very challenging thing to do, and this is the one that I think a lot of the energy is being focused on right now. There's also weight of evidence, where you can build confidence by combining predictions with existing biological data. And so, like I said before, and I'd like to reiterate again, traditional methods and NAMs are meant to be and should be used in a complementary way. And so high quality in vivo reference data is needed to train, test, and validate models. You can't know how a NAM is working, you can't run a NAM or generate a NAM or create an in silico model if you don't have good data to run it with. Modelers need to work with traditional toxicologists to create methods that are biologically relevant. You can create the cleanest, simplest, most cheap or most complex model that you want, but if it's not actually answering a biological question, it's not going to be useful for regulators and it's not going to be useful for most purposes. So you need to work with traditional toxicologists to make sure you're actually answering the right question. And on the other hand, NAMs can be helpful for in vivo modelers, as NAMs can give important context to in vivo results. So you can take in vivo results and make them more human relevant. So you can use Monte Carlo simulations or PBPK modeling or other various methods to model human population variability or create demographic specific results that you couldn't get with just animal studies. So targeting the most sensitive populations that we have. NAMs can also be used for early stage screenings, helping to focus test methods and reduce the number of animal tests that are needed for follow-up studies. So in this slide, we present a timeline of select milestones regarding the regulatory use of NAMs. And for time and for the sake of your sanity and my sanity, I'm not going to go through all of these right now. But what I do want to highlight is that I'm sure many of you are here because of the recent 2025 announcement from the FDA and NIH on the prioritization of NAMs and the movement away from animal methods. And what I want to show here is that while maybe this announcement might be new or it's gotten a lot of publicity, this is not a new concept. There have been decades of documents from US agencies as well as international agencies working towards the regulatory use of NAMs. And so this timeline is very US heavy. I will say that. I'm from a US based company, but you have documents from the Interagency Coordinating Committee on the Validation of Alternative Methods, or ICVAM. You have individual documents from US agencies like CPSC and FDA and EPA and NIH. You have agencies working with the OECD publishing guidelines on how to use both specific NAMs as well as NAMs in general. And so if you would like to read more about this, I recommend you check out the 2024 ICVAM reports. It is this little green circle on the far right where it says ICVAM validation, qualification, and regulatory acceptance of NAMs, as well as I have a link out to it. They've done a phenomenal job of detailing the status of NAMs as they are right now, as well as working towards the future of regulatory acceptance. So that timeline was a more general breakdown of NAMs, including the in chemico, in vitro, ex vivo, and we want to talk about in silico approaches. And so this is by no means a comprehensive list, but I just want to talk about some general trends that we have for acceptance of in silico NAMs. And so during our word cloud, a lot of people highlighted QSAR, which I appreciate because QSAR models or quantitative structure activity relationship models and Redekop methods have widespread regulatory acceptance for screening and filling data gaps. So if you build a high quality QSAR, then you can use those for different regulatory purposes to help fill in those gaps. We have physiologically based pharmacokinetic models or PBPK models that are used in regulatory submissions to predict drug-drug interactions, tissue concentrations, and other pharmacokinetics. So PBPK models are gaining steam. There's in vitro to in vivo extrapolations or IVIVE that can be used in submissions to put in vitro results into an in vivo human relevant context. So there are emerging trends, and you likely know this, but machine learning and artificial intelligence are more than just buzzwords of the day. This is something else that has had decades of growth within the field of toxicology, and they can be used to predict complex toxicities with greater accuracy. We're working on predicting more and more complex toxicity every day, and we are working towards getting regulatory acceptance for these methods. Some more complex in silico tools that my colleagues work with are things like the CATMOS modeling suite or the Sarah ICE models, which are currently working towards regulatory acceptance under different guidelines for acute oral toxicity and skin sensitization, respectively. And those of you who have worked with regulatory agencies likely know that different agencies have different requirements, and that can be even within one country, not to mention across the globe, right, internationally. And so there are ongoing efforts for global harmonization of validation protocols and data standards for these in silico NAMs. And since I'm just giving an introduction, I'm not going to go too far in depth into actual guidelines and things like that. But for resources, if you want a list of NAMs that are accepted by the US government, including in silico and beyond, you can look at the NICEDM webpage linked here to see methods submitted in the EU tracking system for alternative methods toward regulatory acceptance. I linked out here, and then pending interest, we will continue to add resources for regulatory guidelines to the webinar supplementary materials. And more specific case studies for these different methods will be covered in next week's webinars. Something I do want you to consider, though, as we walk through the different methods and different in silico tools, is really know that not every method or model works for every purpose. You need to balance what is the pro of making your own model or working with somebody making your own model versus using an existing one. What is the end goal? What is the level of cost for these different tools? And you may have heard of this before, but something that's likely going to come up again is context of use. Context of use is the manner and purpose of use for a particular method, approach, or application. So always keep in mind what toxicological endpoint is the method testing for, and what, if any, regulatory need does the method address. You'll also want to consider the applicability domain of a model, also known as the domain of applicability. And that is the chemical or biological or parameter space in which the model's predictions are considered to be accurate. And this is widely used as a mathematical cutoff, but I want you to think even beyond just that cutoff of just if you have a model that was developed primarily by people in the environmental toxicity space, you're not really going to want to use it for drug development, right? So make sure that you are understanding the context in which a model was developed and the parameters that were fed into that model and how it might apply to your own purposes. So please model responsibly. And then my final slide before I kick it off to Brad is just a quick in silico NAMs overview. And so in silico NAMs use computers to predict chemical properties, biological activity, potential toxicity, and help eliminate or at least reduce the need for physical experiments. Models and algorithms can predict outcomes based on chemical structure and existing data. These tools can be considered as an analytical layer that help interpret and link data from in silico and in vitro assays. So they can take the different NAMs that we have and bring them together and link them in ways that they couldn't if they stood alone. Or in silico NAMs can help rapidly screen and prioritize chemical and fill data gaps. And they can be widely used to support the 3Rs initiative and are a key component of integrated approaches to testing and assessment, or IALE. And so with that, I think I'm about at my time, which works. And so I'll hand it over to Brad. Brad, I will stop sharing if you want to share yourself.
Thank you, Victoria. All right. Does everybody see my slide?
Yes.
Thank you. All right. Again, my thanks to Victoria. As Kyle mentioned, my name is Brad Reisfeld. I'm a senior principal predictive toxicologist at Inotiv. What I'd like to do now is to move into part 2, now that we have a very good introduction to NAMs and in silico NAMs. Here in this section, I'm going to give you a brief introduction into some of the in silico methods that are going to form the framework for this webinar series. So in future webinars, we'll be presenting case studies that showcase these methods and provide details about the implementation and software. But for the present time, I'm going to give you a little bit of background, a little bit of knowledge about how methods work, what the inputs and outputs are, to give you a sense of their purpose. So the methods we're going to be talking about today are quantitative structure-activity relationships, or QSARs, read-across, physiologically based pharmacokinetic modeling, or PBPK, in vitro to in vivo extrapolation, or IVIVE, quantitative adverse outcome pathways, or QAOPs, molecular modeling, and finally, artificial intelligence, machine learning, and deep learning. That's an awful lot, and there's going to be a lot of material, but again, we will be re-emphasizing those materials during the case studies and you'll have access to the slides and other materials to refresh your memory. But before diving into the materials, we'd like to get a sense of what your current state of knowledge is. So what I would like, if you would be so kind as to go back onto Slido and answer the questions related to this In Silico 101 level of knowledge. This will help us understand your state of knowledge coming in to the webinar, and we'll take a similar assessment after we present the bulk of the technical materials. I promise this is not going to be graded. It's not going to go on your permanent record, so please fill these out to the best of your ability. I'll give you about a minute or so. So these knowledge-based questions are to help align with the learning objectives for this webinar. And hopefully, whether you come in knowing the information now or whether you learn it over the next 20 or 30 minutes, you'll come away from the webinar with that level of knowledge. So give you maybe 20 more seconds. All right, so let's begin to review some of the current level of information. And the first question had to do with hidden layers. And the question was, a hidden layer might be part of what type of in silico model? Now, I'm not going to share the answer at this point. I will at our second assessment, but this gives you a sense of at least the level of knowledge at this point. The majority of you associate hidden layers with deep learning. Next, we talked about chemical analogs. And here we see that the vast majority of responses indicated that read across was the relevant in silico method. In terms of getting internal concentration versus time profiles, the majority of people responded that PBPK modeling was the tool of choice. And in terms of quantitative AOPs, the majority of responses pointed out that key events were part of quantitative AOPs. Finally, in terms of a docking analysis, response indicated that molecular modeling was the most appropriate in silico method. So as we go through the material, if you didn't respond necessarily in the way that you wanted, you'll have a chance to refresh your answers in a post survey. So let's go into our first method, and that is QSARs, or quantitative structure activity relationships. And as the name implies, QSARs are intended to predict biological activity based on structure. They're going to be able to answer questions like, will a chemical show low, medium, or high toxicity for a given endpoint? A question like, to what extent will a structural modification affect the toxicity? So QSARs rely on having a set of chemicals for which the biological activity is known. It also relies on what are called chemical or molecular descriptors. These are quantities that can be derived or calculated ahead of time without the need for experiments. And on this slide, I've shown a few example molecular descriptors: molecular weight, polar surface area, number of aromatic rings. There are actually thousands of different molecular descriptors that can be used. And what happens is then through the QSAR, we're able to take a new chemical and predict its biological activity, either as a numerical value or as a classification or a categorical measure. So here again, inputs to a QSAR, at least for development, are molecular descriptors and these measures of biological activity. When you're actually using a QSAR, generally all you need to input is the structure of the molecule, and the output then would be some measure of the biological activity, whatever that is for your particular application. So on the right-hand panel, there's an indication of the information flow. As I mentioned, chemical structure leads to molecular descriptors. We use training data with known activity, and this then through a statistical or mathematical model gives us the ability to make predictions. And these models could be machine learning, could be regression, statistical models, that kind of thing. Our next method is read across, and read across is used to fill data gaps by transferring information from similar molecules. And we generally call these similar chemicals chemical analogs. And so the information of interest could be some molecular property that we're lacking. It could be something having to do with the toxicity or the activity. But the tricky question here is, what does similarity mean? Does it mean something having to do with structural similarity? Does it mean that they're similar with respect to some kind of particular physical property? Well, that depends on the application and the mechanisms that are at work. So for instance, if we were interested in estimating metabolic clearance rates, similarity might mean that the molecules possess certain functional groups that interact with the enzyme, and that those should be similar within the analogs. It could mean that molecules are known to share similar metabolic pathways. If we're interested in something like whether a chemical can diffuse across the blood-brain barrier, similarity might be things like charge or polar surface area. So once we have an idea of what similar means, we can search for these analogs and look for those that have relevant activity. This is often a challenge. There's various databases that can make this task a little bit easier. So again, inputs to read across are generally things like chemical structures, descriptors, and various properties of interest. And what we get out of our read across is an indication of this unknown property, as well as hopefully a measure of uncertainty or confidence in the result. So we don't build a mathematical model in this case, but we use the value from the closest analog that we find. Alternatively, we can interpolate or extrapolate based on several analogs. All of this requires some expert knowledge, and there's some types of software that can also help us for read across. Next method is physiologically based pharmacokinetic models. And here what we're doing is we're predicting the tissue-specific absorption, distribution, metabolism, and elimination, and concentrations of chemical species in the body over time, given a certain exposure and route of administration. Just as a note, most of you probably know that pharmacokinetic and toxicokinetic are often used interchangeably. The former often implies drugs, while the latter usually implies toxicants. I'm a little haphazard in this, but I'll generally be using PBPK or pharmacokinetic even when I mean a toxicant. So we can answer questions using physiologically based pharmacokinetics, like what is the maximum concentration of a toxicant at a site of action, for instance, the liver? What is the half-life of a chemical in, let's say, adipose tissue? So in PBPK modeling, we divide the body up into a number of tissue compartments, as are shown in the left here, and we connect them through blood flows. We associate them with physiologically relevant volumes and flow rates. And through this physiological and biological association, we can do extrapolations, or at least in theory, we can do extrapolations by upscaling volumes, by using different what are called allometric relationships. We can extrapolate across exposure routes. And the nice thing is also that we can include mechanistic relationships, such as complex metabolism, or things like mechanistic models for transporters. So there's a lot of parameters that are required for at least building a physiologically based pharmacokinetic model. We need these physiological parameters. We need chemical specific information, physicochemical information like partitioning, log P, things like that. We need metabolic information, Vmax, Km, or things like overall clearance rates. We often also need a rich set of in vivo data so that we can parameterize the model and find unknown parameters. But the nice thing is, once we have a validated model, we can make predictions of these tissue-specific concentrations and ADME type measures within tissues of interest, target tissues, routes of exposure, things like that. And on the right shows a little bit of the information flow through a PBPK model. Let's talk now about something that's somewhat closely related to PBPK modeling or uses PBPK modeling, and that is in vitro to in vivo extrapolation. The idea here is that we want to translate in vitro assay results into predictions that are relevant for a whole organism. So it aims to answer a question like, what is the human equivalent dose that corresponds to an observed in vitro response? So how does all this work? Well, essentially, we look at our in vitro dose response curve, like shown on the left here. We pick out some threshold of biological response and find out what the corresponding concentration is. We then use that concentration within a PBPK model as the basis for determining the exposure. So what we essentially do is, if we have an in vitro assay that's associated with a particular tissue, say hepatocytes, we could associate that concentration with the level in the liver compartment of our PBPK model. So that gives us a tissue concentration. Then what we do is we run the PBPK model in reverse to find what exposure or dose level led to that internal concentration. That process is sometimes called reverse dosimetry or dose reconstruction. So what do we need? We need all of the same things we required for the PBPK modeling. Additionally, we need our in vitro bioactivity data, ideally dose response information, and some idea of exposure as well. What do we get out of that? Human equivalent doses are a common output, as well as bioactivity to exposure ratios. Shown on the right is, again, this workflow or information flow going from in vitro assay results all the way to, let's say, an HED. So before jumping into our next topic, which is quantitative AOPs, I'd like to get just a very quick indication of how familiar you are with AOPs in general. If you could just give a quick thumbs up if you have experience with AOPs, and maybe a thumbs down if you have no experience, that would be very helpful. A lot of thumbs up. Nobody wants the thumbs down today. Okay. Thank you. So this is very helpful. So many of you are already familiar with AOPs. And so like a standard, what I'll call a standard AOP, quantitative AOPs aim to connect a molecular initiating event to an adverse outcome through a series of key events. So our standard AOPs focus on things like biological plausibility, mechanistic understanding, but not necessarily numerical values. They tell us what happens and why, but not how much of a chemical is needed to cause an adverse outcome, or to what extent other factors in the system influence the outcome. So a QAOP takes the same sequence of events and basically overlays key event relationships. These relationships can be mechanistic, connecting to the key events, let's say. They can be semi-empirical, they can be data-based, so utilizing some data acquired through separate kinds of testing, or they can be probabilistic. In the latter case, the idea is that given some key event, let's say key event 2, if we know that's happened, a probabilistic model would say, then given that that key event has happened, what is the probability of the following key event happening? And that's a common way of formulating a quantitative AOP in terms of what's called a Bayesian network. So to construct an AOP, many of you know there's an awful lot involved, including knowledge of these molecular initiating events. But aside from that, we also need to have this quantitative relationship between all of these different key events. And so that requires an added layer of investigation, potentially involving phenomena that occur at multiple levels of biological organization, from the molecules to cells to organs to organisms, all the way up to population levels. The nice thing is, once all that's done, is that we can get quantitative information regarding the transition between these key events. We can look at predicting dose responses. How does the dose or the a perturbation to the system affect the adverse outcome? And they can be used for weight of evidence for looking at the relevance of a pathway. And that information just shows what I just described, where we're looking at potentially multiple layers of organization and these response functions or key event functions that connect our different events. So our next method is really more of a collection of methods, and this is molecular modeling. So molecular modeling aims to predict things like molecular structures, molecular properties, and interactions between molecules and atoms. It can help us answer questions like, what's the preferred three-dimensional structure of a molecule? is my toxicant likely to be metabolized by a certain enzyme or bind to a certain receptor? Can intermediates form when my chemical reacts? Those are all questions that can be answered by different aspects of molecular modeling. In terms of molecular modeling, in terms of application to toxicology, I think there's 4 main areas: computational quantum chemistry, molecular mechanics, molecular dynamics, and molecular docking. So our computational quantum chemistry, we can use to predict molecular properties, energetics, and interactions. And you can actually look at reaction pathways and things like that through certain kinds of CQCs. Molecular mechanics provides a relatively quick way to determine an optimal structure for a molecule, or the most favorable energetically structure for a molecule. Molecular dynamics looks at motions, and it can simulate both atomic and molecular motions. For instance, we might be interested in the motions of a transmembrane protein in a lipid bilayer, and molecular dynamics can look at that kind of a case. And finally, molecular docking can make predictions related to the interactions of a ligand with an enzyme or a receptor. So if we wanted to carry out, let's say, a molecular modeling study, we'd of course need the structure for our chemical, which we can get through our molecular mechanics. We also need a structure. Generally, we need a structure of a protein if there's some kind of interaction, an X-ray crystallographic representation, or we can even use something sort of like read across that's called homology model. We need to know something about force fields or quantum mechanical parameters, and those are usually handled by our software. So the things that we can get out are molecular conformations that are energetically optimized. We can look at different properties of the molecules. We can look at protein ligand binding poses and affinities. So if we focus on docking, what happens is that the docking software will essentially look at putting our molecule into the active site of the protein and trying to minimize the energy and avoid steric hindrances. It then sets a score for each of these, what we call poses, based on shape, complementarity, electrostatics, things like that. And then the score can be used later to find the most likely pose and the corresponding binding affinity. So our final topic is a very broad one. And I think we all know about AI through at least large language models and ChatGPT and OpenAI. What I want to point out is really these, what I've termed them as AI, machine learning, and deep learning, aren't really 3 separate things. Artificial intelligence is this larger umbrella under which machine learning falls. There's other topics under there as well. And under machine learning, we have something called deep learning. And we use this somewhat long name for the topic because you'll see in the literature different types of terminology. Really, the thing that we exploit, regardless of the method, is that these approaches can be used to identify and quantify patterns in large data sets. They can provide highly nonlinear relationships that are consistent with our data and make predictions and help answer mechanistic, predictive, integrative, prioritization questions. And on the right-hand side, you can see some of the areas in which these areas of this in silico approach can be applied. So to construct one of these models, we normally need a large training data set. And large is somewhat dependent on the application of interest. Typically, we'd need chemical structure information, bioactivity kinds of data. Along with the base data set, we also need data for validation of our model. We'll typically need chemical descriptors or some way of characterizing a model that can be related then to the bioactivity. And what we get out are predictions of toxicity. We can get out things called feature importance, and that tells us which of all these chemical descriptors had the most influence on making the prediction regarding the bioactivity. Generative models can give us synthetic data, which can be useful. And a necessary part of these is also some metric for how good the model is, its accuracy, sensitivity, and selectivity. So on the right-hand side, I've shown a deep learning model, which is a kind of a neural network. And neural network has different layers associated with it, an input layer, some hidden layers, and an output layer. And we have these hidden layers because they're really unobservable as opposed to the input and output. And out of this array of artificial neurons, which are very simple in and of themselves, we can generate predictions. And as toxicologists, things like whether something's toxic or non-toxic, or more fine-grained differentiations of quantitative levels. Well, let's go ahead and reassess your knowledge now that I've given you an awful lot of information in a very short time. So please retake the quiz or poll. All right. So let's take a look. Awesome. So we see here that in terms of hidden layers, as we just saw, hidden layers are typically a part of deep learning, 100% response rate. Awesome. Chemical analogs being associated with read across, 100%. Again, excellent response. Kernel concentration versus time profiles are typically associated with PBPK modeling. They could be predicted through machine learning or read across, but really one of the sole purposes of PBPK modeling is to determine these internal concentrations. For quantitative AOPs, 98% of you said that key events were a key component, and that is certainly true. Descriptors we would associate more with QSARs. Docking is a form of molecular modeling, so good job on that. Docking can also be performed using aspects of deep learning and machine learning, but primarily it would fall under the purview of molecular modeling. So one other ask from us has to do with the relevance of what we've shown you today in terms of your own work or just your personal interest. So if you'd be so kind as to rank how these in silico methods fit into either your professional life or your intellectual curiosity, that would be awesome. So we have a lot of interest in QSARs and read across, but there's a pretty good balance across the field. So it looks like in terms of your level of interest or relevance to your work, pretty much all of the methods that we've discussed today have some relevance. So I really appreciate your input on that. So just summarize, in part one, Victoria noted that in silico approaches are really valuable in complementing in vitro and in vivo studies. They're able to enhance the evidence base for chemical hazard characterization and risk assessment. She also showed a timeline and other information that indicated they're expanding use across multiple sectors with growing acceptance for certain regulatory applications. And then finally, I kind of fire hosed you with a lot of information on in silico methods, including required inputs, generated outputs, and overall information flow. So overall, we sincerely hope that you learned something today, and we really appreciate your attendance and participation. So thank you very much. Are there any questions?
We do have questions, Brad. We will get started right now. Thanks, both Brad and Victoria, for your question or for your presentations. Those were really nice theoretical basis for a lot of in silico models, and we can look forward to next week. The webinars will show us how the concepts that were talked about today can be illustrated with case studies. All right, so let's see what we have in terms of the questions. What role does external validation play in QSAR validation? How can reviewers objectively assess the performance of such models? And that can, either of you can take that question.
I think that's a great question, and I think part of the validation involves really a close examination of a couple of the factors that Victoria mentioned of range of applicability and the uses that the QSAR is going to be used for. Taking a close look at what data were used to create the QSAR, as well as the rigorousness of the evaluations that were done, can be good, as long as looking at critical metrics for QSARs and not just looking at a scant test set. Worst case, that the test set is part of the training set. So I think, external validation, or at least critically examining the ways in which the QSAR were developed, are good pointers towards the validity. And assuming that the range of usage is going to be within that same sort of spectrum of chemical class or applicability domain. Victoria, do you have anything to add or?
No, I think you summed it up better than I ever could. So.
Okay, great. Thanks, Brad.
Yeah, thank you guys. So I'll take up the next question here. We've got, I think, keeping it in scope for the kinds of things that we talked about today. I see a question that says, all these methods are fine for research, but to what degree are their applications accepted for regulatory use? And I know that's a lot to think about. Are there limitations to how methods can be used in that context? And I know we'll get to an opportunity to talk about this more as we go through this webinar series, but perhaps just kind of a brief thought there.
Well, I think Victoria mentioned some of these in silico tools in particular as being used in a regulatory context, like the QSARs, like PBPK modeling, like read across. So many of them are more than just research methods, but are being used. FDA, for instance, in the portfolio submitted with new drug application can accept PBPK modeling results. So there is, I think, for some of the other methods, their usage may not be as widespread. Some of the QAOPs are at least designed with a regulatory purpose in mind, whether it be in ecotox or in human health risk assessment.
Thank you.
And I will just add, I think I mentioned this in my slide, but there are resources for where you can find various regulatory acceptance criteria, as well as OECD publications on what are they looking for with QSARs and PBBK, and we can link those in our additional resources so you can find them yourself or track down various accepted in silico methods through the ones that we linked in our slide. So it's a really good question. It definitely varies across method, and we'll try to help you navigate that.
Thank you,
Okay,
Victoria. Those resources would be really helpful. Sorry, Christy.
no worries. Thanks both. The next question I've pointed out here is, what resources for understanding and using AI tools can you recommend for a toxicologist without any prior experience in this area?
We're starting to put together, or we have put together, an additional resources document that I think is now posted. There is a section in that that has information on applications of AI, machine learning, and deep learning. There is increasing coverage of those topics in references related to computational toxicology. Some of the latter or recently published books have had a little bit more coverage of those areas, and some of those might be able to give you a basic grounding in some of those things. We have, as part of this additional resources, we have a section devoted to computational toxicology in general. Sadly, I don't know of any super accessible resources for novices, unfortunately, that are focused on toxicology, but I think those might be good starting points, the resources I just mentioned.
Anything to add, Victoria?
I was going to say that I think we are also hoping with this webinar series to be that access point. So if this is something that you find useful, or if there's certain topics that you would like to cover more than others, please let us know. AI definitely is a black box in a lot of ways, and it's hard to navigate. So this webinar series is trying to be that entry point for most folks.
And webinar 3 will cover a case study for the use of AI, machine learning, or deep learning, and walk through at least a published case and go into some of the details of how using that technique was useful in answering questions relevant in toxicology and risk assessment.
Well, I see that we've run over here. Krista, do you think we still have time for some more
I think we
questions?
can go ahead and continue asking questions. Folks can jump
Great.
off if they want to. Yep.
Great, thank you. So I see a question here about potency. Of the methods that you presented, Brad and Victoria, Which of these methods have the most use for potency prediction?
I think that provided the data set is rich enough, a QSAR could be a reasonable method of doing a potency prediction. What we like to do, and I didn't really mention it, is that to the extent that you have a mechanistic understanding of the toxicity, that can help inform a QSAR such that you use descriptors that are relevant to that mechanism to help build your QSAR. So there's some biological underpinning. So my recommendation, or at least my initial thought, is that, again, provided you have enough data to both tune or train a QSAR and to test it, I think a QSAR could be used for potency prediction.
Okay, thanks.
Thank you.
All right, so I'm going to ask this question. I think they're mixing up different kinds of models, but we'll see what you guys say in response. Has anyone already taken the PBK modeling to the next step by connecting internal concentration estimates to the effect prediction, such as X concentration could cause Y effect in Z organ?
Yes. And that would be really combining the PBPK with a pharmacodynamic model. And so we didn't talk specifically about pharmacodynamic models. That's where we look at, so pharmacokinetics is what the body does to the toxicant, and pharmacodynamics is what the toxicant or the chemical does to the body. So looking at that latter element of pharmacodynamics, even in using something as simple as a dose response curve, those two could be linked together so that the output, a tissue concentration, can then be cross-referenced to some outcome of interest. So again, the combination of pharmacokinetics and pharmacodynamics, I think, is what the person was referring to, and that is something that's relatively commonly done.
Okay, great. Thank you.
All right. I have a question that I see here in the chat that I know at least a few people personally would love an answer to. How would the proposed IVIVE approach address inhalation dosimetry, especially the proximal to distal distribution from nasal to pulmonary regions? I'm seeing this and I'm thinking about something like MPPD modeling, but I'm curious what your thoughts are, Brad and Victoria.
That's my thought as well, is that a more sophisticated PBPK model than the one that I showed with inhalation is a common route of exposure. There are some PBPK models that include a computational fluid dynamics portion that can look at deposition of aerosols and particulates in the airways, looking at spatial variations, looking at the effect of mucus, things like that. So I think if we have a validated model that gives us that level of granularity, that might help answer questions related to the linking the IVIVE or the in vitro information to an external dose.
Thank you.
Does that help?
Yeah, I think so.
Mm-hmm.
It strikes me as not necessarily a 101 question. Our audience is getting advanced already.
It's a really interesting topic, though.
Yeah.
I love it.
So there are a few questions like that in here, like where we're getting into sort of different similarity metrics and details about PBK modeling. and rejection rates of read across or read across watchouts, then I think what I'd like to do is say that if you want to come to the next couple of webinars, we'll be delving into some of these topics in more detail, and either the questions will be answered or you can ask them again, and we'll be able to kind of get into a richer discussion around some of these topics. I also wanted to mention that some of the questions are being provided with typed answers. So if you asked a question and it didn't get answered orally, it's in the little answered tab. You can see the typed answer. So be sure to check that out. And so I think because we're over time, I want to go ahead and respect our speakers' times and all of your time and close out the webinar. So I want to thank you all, and I apologize that I don't have my camera on. I'm traveling internationally and I don't have a really good, reliable Wi-Fi signal, but many of you know what I look like already, so that's okay. So thanks again, all of you, for your attention. Thank you to Brad and Victoria for sharing your knowledge with us. And we hope you will join us next week for webinars number 2 and 3. Thanks again for all of your attention, and hope you all have a great rest of your day.